Hello everybody, welcome back to another video. This one is going to be focusing on a little bit of cellular respiration as a whole activity, but we're going to spend a little bit more time focusing on anaerobic activities, including glycolysis, which is the first step of both cellular respiration and anaerobic activities. Okay, so let's talk a little bit about cellular respiration. If you took just general biology last year, you probably learned that it was just the process of breaking down our food. Maybe even you learned a little bit more than that, depending on your teacher. But what now we need to understand is that it's a very highly ordered process that cells do in order to acquire energy from the food, the nutrients that we eat. The one that we focus on for cellular respiration is glucose, but we'll talk a little bit about how other things are also broken down, like proteins and fats, later on. And the goal is to get ATP. And that's because ATP is going to be able to be used in more various contexts. So instead of using the energy that is stored in bonds and glucose and coupling that with other reactions, instead we funnel it into a molecule called ATP, the energy, to ATP in order to use that energy for lots and lots of different contexts. We'll see what that means in a second. But first thing we need to understand is that ATP is going to be used for cellular work. So the goal is to break down glucose. Glucose is a 6-carbon molecule. We completely oxidize it, meaning that we completely break it down into individual carbons that are going to be released as CO2. When we break down those bonds, when we break down glucose, we are able to store that energy into ATP, which can be used for other processes, things like transcription, translation. For plants, they do photosynthesis. Anything that is an anabolic process probably requires ATP or a different nucleotide triphosphate. So we talk about ATP, but I did tell you guys before that you could also potentially have something like GTP, CTP, or even UTP. Okay. So let's look at the formula for cellular respiration. I think this is important. The entire course or the entire unit is all about cellular energies. And so energy is naturally tied with chemistry, so we need to understand these formulas. So you should remember from photosynthesis that we had energy, CO2, and water as inputs, and then we got out glucose and oxygen. Well, if you know that, then you basically know cellular respiration because you just flip it around. So instead, now we start with glucose and oxygen, and we are going to produce carbon dioxide, water, and energy. Now this energy is not sunlight, this energy is now ATP. The main thing that I want you to take away from this slide is this idea that CO2 is the complete oxidized version of glucose. So we completely oxidize glucose because these 6 carbons are bonded together. The 6 CO2 molecules, they're not bonded together. We just have 6 individual molecules, and those are each 1 carbon each. So this is really important. I like to learn pathways by counting carbons. That is the easiest way to remember inputs and outputs for me. And so that's the way that I tend to teach it. Okay, now there are a couple of new players. So if you watched photosynthesis, then you should be aware of a molecule called NADPH. NADPH does not show up in cellular respiration. Instead, our electron carriers are NADH and FADH2. Just like NADPH, they are reusable. Their whole job is to take electrons and then drop them off. So NAD will accept an electron, as you can see in this image right here, and then it will go and deliver that electron somewhere else. Where it is, we will get there when we get there, but I want you to understand its whole job is to just shuttle electrons around. So NADH is very closely related to NADPH, which I'll show you on the next slide. FADH2 is a different electron carrier. It is important that you know it. The structure is really not that important. Just understand that they are going to be reused and they are going to deliver electrons around. Okay, so let's look at the difference between NADH and NADPH. The difference is really just this phosphate group. And so that's fine if you want to remember that it's a different phosphate group. The way that I remembered it when I took AP Biology is I remembered P for photosynthesis. It doesn't stand for photosynthesis, but it helps me remember that NADPH is for photosynthesis, and then NADH was just the other one that was for cellular respiration. Okay, so Just like we talked about in photosynthesis, we need to understand where cellular respiration is happening, because there are so many moving pieces that if we don't know where things are happening, it'll be very easy to get lost. So hopefully you know that cellular respiration is going to happen in the mitochondria. Now, we need to put a little caveat on that, because when we talk about glycolysis, you should remember from class that glycolysis happens in the cytoplasm. Everything else for cellular respiration is going to occur in the mitochondria. And you should also remember that the mitochondria, because it was part of the endosymbiotic theory, right? So it was likely phagocytosed billions of years ago, so that's why it has a double membrane. And both parts here are very important. Now, if the mitochondria was once its own free living cell, then it's likely to have something that resembles cytoplasm, and that's basically what the matrix is. The matrix is essentially the cytoplasm of the mitochondria. We don't call it cytoplasm because we don't consider the mitochondria its own cell anymore, and cyto means cell, but I do want you to understand that it's a very similar thing. Then we have these things called cristae. Cristae are just folds in the membrane, so it is going to be there to increase surface area. So if you remember from our cell unit, Unit 2, then you remember that cells are often trying to maximize their surface area to volume ratio in order to dissipate heat, but in this case it's to really maximize the space for the electron transport chain, which we're going to talk about in another video. And then, very well named, is the intramembrane space. So between the two membranes, the outer and the inner, there's going to be space there. And so that's why we call it the intermembrane space. Okay, so today we are going to be talking primarily about anaerobic activities. Aerobic means that it requires oxygen. That is not what this video is all about. This video is about anaerobic activities, meaning that it does not require oxygen. So I come back to this graph over and over and over. I wouldn't call it a graph, actually. I call it a figure. And I want you to understand that this is showing the path that glucose can go. So naturally, glucose has to be split into a smaller molecule. Let's just say that it only turns into pyruvate for simplicity's sake. Now, the fate of pyruvate can vary. If we have oxygen, then it's going to typically go this route. And this is the aerobic respiration route. It's going to go through the Krebs cycle, go through the electron transport chain, and then make a whole bunch of ATP. This is all going to happen in the mitochondria. But if there's no oxygen around, then pyruvate is basically forced to go the other route, which is the anaerobic route, and that takes place in the cytoplasm. So that is where we are going to see fermentation. Now, humans don't do all types of fermentation. We do lactic acid fermentation, which we'll talk about in a little bit. But I do want you to understand that if we have no oxygen around, we're going to go the anaerobic path. By the way, we consider glycolysis also anaerobic. So it is also going to happen in the cytoplasm. So let's talk about glycolysis. It is the first step of anything dealing with glucose, essentially, at least for our course. So glycolysis is going to occur in the cytoplasm. Here's the way that you can remember what glycolysis is. Lysis means to split. We've talked about that before. We saw it with hydrolysis, using water to split things. In this case, we are splitting a glucose molecule. Glyco means pertaining to a sugar. We've seen it before. Glycolipids are sugars attached to lipids. We've seen it with glycogen, which is a chain of glucose molecules, and now we're seeing it here with glycolysis. So we split a glucose molecule. Now, thinking about carbons again, we have 6 carbons in glucose, and then we split it in half to make 2 3-carbon molecules. So the same number of carbons here. That will become really important when we start counting CO2s. Now, almost all organisms do glycolysis. This is really important from an evolutionary standpoint, because the Earth, billions of years ago, did not have oxygen. And so this is a way that organisms could evolve and produce their own energy when there wasn't oxygen around. Now, it's not particularly efficient, but it does get a little bit of ATP, and it can do it without oxygen. So this probably evolved first, before the Krebs cycle, before the electron transport chain. Okay, so as we are looking through these pathways, from glycolysis to the Krebs cycle to the electron transport chain, I'm going to let you know that there's a lot that we're not showing you at this level. Now, if you go take biochemistry, you'll see a lot more glycolysis, for example, is 10 steps, 10 different enzymes, and there's a lot of cool chemistry that's going on. But for our purposes, for AP biology, it is important that you understand two major steps of glycolysis. The first part is the energy investment step, which is the first half here. Above this dotted line is the energy investment step. So just like you could invest in money into something like Google, you invest it with the hopes to get more out. So in this case, we invest 2 ATP. When we split glucose into 2 3-carbon molecules, in this case this is G3P, and yes, that is the same G3P that you see in the Calvin cycle. So we invest 2 ATP to get this split done. But now this G3P can be converted into our final product of glycolysis, which is pyruvate. This returns 2 ATP back for each one. So I spend 2, as we can see here, but I get back 1, 2, 3, and 4 ATP. This is going to be netting 2 ATP. So glycolysis does produce ATP, but when we start seeing the electron transport chain, we're going to see that it's very minimal in comparison to what you can get if you have oxygen present. So this second phase, we call it the energy harvesting step. I also will commonly call it just the returns, because if I invest something, I want my returns, right? And so this is the energy harvesting step. It is where you get your energy back plus some. So overall, glycolysis, you spend 2 ATP, you get 4 back, so we net 2 ATP. There are also other important molecules here that you should know. You should know that we are getting 2 NADHs per glucose. This is really important when we start talking about the electron transport chain later on. Now, there is a term that you will hear related to glycolysis very often called substrate-level phosphorylation. This is where an enzyme passes a high-energy phosphate to an ADP to form an ATP. So in this case, we've seen this term substrate before if you watch the enzyme videos or if you're in my class. So a substrate is going to enter the enzyme at the active site, and then in this case, this enzyme is going to pass this phosphate group, this high energy phosphate group, to an ADP. And then that ADP now has an additional phosphate, so instead of being a diphosphate, it is a triphosphate. And now this product is going to be the substrate minus 1 phosphate plus the ATP. Now, where this phosphate comes from, well, you might have noticed before that G3P has a phosphate. The very first step of glycolysis is to add phosphates. That's what those ATPs are used for in our investment phase. They're to add phosphates to a glucose molecule. There's a lot that we're not showing, but do understand when we say substrate level phosphorylation, it's just moving a phosphate group to an ADP. That's going to be in contrast to oxidative phosphorylation that we will see in a different video. Okay, so let's assume that we do not have oxygen. We're going the anaerobic route. What happens? Well, pyruvate can really only go do fermentation. So what is that? Fermentation is a process that is going to produce a limited amount of ATP, and it's not really doing it directly. We'll see what I mean by that in a second. It's not really producing ATP. Instead, it's going to be recycling NADH. So let's go see that. So there are 2 routes that it can take. So pyruvate can either be converted into lactate or it can be converted into ethanol. Now the key part here is I don't see ATP as a product on either of these routes. That's important to know, that fermentation itself is really not directly making ATP. Instead, what we make is a very important, or we regenerate NAD+. Now that NAD+ can be used elsewhere, and I'll show you now where that is. So in the case of lactic acid fermentation, that NAD can go back to glycolysis. And now that NAD can be converted into NADH, yay, but also the process of glycolysis nets to ATP. So while fermentation didn't actually yield any ATP, it did yield NAD, which can go back to glycolysis to make NADH. if we don't do this, we'll quickly run out of NAD, which will shut down everything. So that is why fermentation happens, is it's a short-term solution to make just a little bit of ATP by recycling NAD and going back to glycolysis. Now, what organisms do this? Well, I do it, you do it, bacteria do it, and they do it when there is no oxygen available. So think about when you are sprinting as fast as you can, your legs will typically burn. And that's because you are using up all of the oxygens that are stored in your cells pretty quickly. And so your body is forced to make a short-term supply of ATP, and they will use lactic acid fermentation. That is why your legs burn. It is lactic acid. Now there is a different type of fermentation called alcoholic fermentation, which I don't do, and you don't do either. So alcoholic fermentation is pretty similar in that the goal is the same. It's to recycle NAD to go back to glycolysis to net 2 ATP, but there is an extra step here. And this is where counting carbons can be really important. Hopefully you remember that pyruvate is a 3-carbon molecule, so we don't change the number of carbons in glycolysis. We start with 6 in glucose, we end with 6 with 2 3-carbon molecules of pyruvate. However, I'll tell you that ethanol is 2 carbons, which means that we have changed our carbon number. We went from 3 in pyruvate to 2 in ethanol. And so where do those carbons go? Well, they leave. And you're going to see this anytime we change carbon numbers, that means that carbon had to leave, and it leaves in the form of CO2. So we say bye to the CO2 and we never think about it again for our course. Now, the organisms that do this are going to be bacteria, plants, and yeast. Now, yeast is the one that most of you guys probably care about, because that's the stuff that makes alcohol that people drink. I don't recommend it. It tastes bad, and it's not good for you. But that is how alcohol is made. In fact, yeast will produce so much alcohol that it will kill the yeast, and then they'll sink to the bottom, which I find a little gross. So that is anaerobic respiration, that's fermentation, that's glycolysis, and an introduction to cellular respiration. So I hope that you learned something and that you're feeling a little bit more confident for the exam. I'll see you soon.